Hey guys, this is Mr. Farrell. You need to watch this video in order to fill out your notes for, let me get on the right classroom, that might help. To fill out your notes for week 5. This is our passive transport notes. So we are talking about we've talked about last week, we talked about the different parts of the cell. We talked about the organelles. Remember, organelles, these are similar to organs in our body. So like your stomach, your liver, your heart, your lungs, those are all different organs and they have different jobs. They look different. And so we should think, okay, they look different. They probably do different things. Just like the organs in your body, the cell has organs. They're not called organs, they're called organelles. They're just tiny versions of organs. And we talked about those last week. So this week, we're going to be focusing in on one of those specifically the cell membrane. So if you remember, the cell membrane let things go in and it let things go out, and it was something we called selectively permeable. That means it chooses or it selects what can go in and what can go out. So we're going to talk about that this week, and we're talking about how do things move in and move out of the cell, because that's really important. Cells need food, cells need water, cells need oxygen, they need to get rid of waste. So it's really important for the cell membrane to work right in order for the cell to function. If that doesn't work right, then the cell's in trouble. So we're going to talk about this week something called passive transport, and we're also going to talk about something called active transport. So if you click into here with these notes, you should be filling these notes out as we are watching the video. So to start off here, we have different parts of the cell membrane. So this is the cell membrane right here. And if you notice, it's got a couple of different things. It's got these yellow guys that have a circle, and it's got two little lines. We call those tails. We call the circle heads. And each one of these things is something different. So, this circle here and the two tails that are with it, that's one thing. We call that a phospholipid. So a phospholipid is a lipid. Remember from last week, lipids are fats. So we have that. And if you look, we also have these pink things, or we can, purple, whatever color you want to call them. These are proteins. Now, last week we said that proteins are, we think about them as, being involved in muscles and building muscles, and you eat that to build more muscle. But proteins do a lot of different jobs. Last week I mentioned proteins are, the example that I use is they're involved in hemoglobin. Hemoglobin is a protein. And what that is it's a protein in your red blood cells that's responsible for carrying oxygen. So that's what helps your blood carry oxygen, is the hemoglobin, that specific protein. Now, if you look, we got different kinds of proteins. This one here, it's got a hole through it. So it looks like something can go through it. And that's exactly what its job is it's a protein channel. So it acts like a doorway. So it lets things go in, it lets things go out. And then we have some other proteins. We have some embedded proteins here. That just means that it's stuck inside the cell membrane. But different, if it's shaped differently, if it looks different, these two things are going to have different jobs, even though they're the same color. Also, we have these carbohydrate chains here, and these carbohydrate chains they act like signals. They receive signals. They are almost like an antenna, and they do various sorts of signaling and receiving signals and sending signals. Now, we call this here. If you look, we have some phospholipids up here, and then we got others that are flipped upside down. So we call this a phospholipid bilayer. And the reason we call it a bilayer is because we got two layers. So bi means two. So we talked about that before. We talk about different word parts. So bi means two. So there's two layers of phospholipids here. Okay, so that's the first thing that we'll talk about. Now, moving on along, we mentioned also last week, and I mentioned it already in this video, that the cell membrane is selectively permeable. it's selectively permeable. So what that means is it allows some things to go through and other things not as much. So the cell membrane chooses what can come in and what can come out. Okay. Now, one thing that we have is something called diffusion. Scoot this over just a little bit more. So diffusion is the process where things will go from a high concentrated, so like you got a lot in a little space, to a low concentration where the same amount is spread out. One really good example to think about is maybe like perfume or cologne, where you have all that stuff in the bottle, and if you open up the bottle, if you're able to like unscrew the top and open it up, it's really, really strong. That's because you have a lot of perfume, a lot of cologne in a little area. But if I take it and I spray it in the room, it may smell strong at first, but eventually it gets to the point where it's not as strong and you may not even notice it. Well, that's because those particles are moving around, and eventually, instead of being in a small, tight area, they kind of spread out. And we call that a low concentration when it's all spread out. So eventually, it's going to get to the point to where you can't even smell, like it'll smell the same throughout, whether it's whether you can smell it or not. So like if we stay with that cologne or that perfume example, when someone sprays the cologne or someone sprays the perfume, it's going to smell really strong right where they sprayed it. Well, that's because all those particles are there, and eventually it's going to spread out and it's going to reach something called equilibrium. So going to reach something called equilibrium. And what that means is those perfume particles or those cologne particles, they're spread out evenly throughout a space. So there's not going to be an area that smells stronger than another. It all like smells about the same intensity. You're not going to just go through a cloud of like really strong smelling perfume and then go to an area where you don't smell it. Like it's going to be the same. Now, I know sometimes, like if I'm sticking with this perfume or cologne example, there may be times where we may not smell it, or you'd be like, well, the smell's gone. Well, it didn't really go away. It's just, it's so spread out that we don't really recognize it or catch it anymore. So moving on right along, next is this idea of concentration gradient. So a concentration gradient, it's the difference between concentrations in a space. So it's the difference between highly concentrated area and a low concentrated area. So like I said, smells are a really good example of this, whether it's perfume or cologne or whether it's someone passing gas, that is a smell. You have, if we're talking about that example, you have, if someone were to pass gas, you have that area where they, right when someone like passes gas, you have that area that's super, super strong. And so that's a highly concentrated area. Well, it's going to move to places that's lowly concentrated and doesn't have the smell. So it's going to spread out just like the perfume. So the difference between that high concentrated area and that low concentrated area, we call that a concentration gradient. But there's many examples of this and stuff that you're familiar with. Like I said, any sort of smell like that. We can talk about concentrations for like food. If we're talking like orange juice, you go to the grocery store and you go in the freezer section and you find like the little can of concentrated orange juice. There's that. There's many examples here. So moving right along, sometimes we have things that are really small. Let me scoot up here first. Sometimes we have some things that are really small, and what they can do is they can actually fit in between these phospholipids. They can kind of just squeeze in between there. Those are going to be really small things like oxygen, really small particles, and they'll just diffuse through the cell membrane here. Others, they're really big. And the only way they're getting through is using some sort of door. They can't squeeze on by. They have to have like a clear way, have like a doorway here. If we have something that's big that can't pass the plasma membrane or the cell membrane, that's going to use something called facilitated transport. And basically all it means is it just needs help. And it's going to need help by carrier proteins. So this is still going from high to low concentrations, but it's just going to need help with carrier proteins. So if we're looking here, all right, if you look, we have the outside of this. We'll say this is the outside of the cell, and we'll say this is the inside of the cell. And if we look here, this is our carrier protein. And so it's trying to point to this, and then these little dots are glucose. So first thing we got to do is we got to figure out the concentration. So if you look here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. We got 12 glucose molecules here. We got 1, 2, 3, 4, 5, 6, 7, 8. So there's more glucose on this side of the cell membrane than there is this side. So I'm going to use my highlighter here. So this is going to be the high concentration. Let's try this again. That's going to be the high concentrated side. And this side is going to be the low concentrated side because there's more things over here. This is where the glucose is stronger. There's more of it over here than there is here. So with diffusion, we're going to go from high to low. Now, for osmosis, if we're talking about osmosis, this is the diffusion of water across a membrane. So it's diffusion, but it's more, it's in a special case. It's for water. And so we talk about this a lot. And later in week 5 work, you're going to have a lab that you're doing talking about water and dealing with water. So water's going to move, its diffusion is going to move from an area of high concentration or low concentration. But a lot of times we don't have just water. So especially in the body, there's all sorts of stuff that has different chemicals or salts or anything like that. So water is going to move in the direction where there is a high solid concentration. So there's a lower concentration of water. So if we think about maybe salt water, okay, like the ocean or something, okay, if it's very, very salty, if it's like 70% salt, well, the rest of it's going to be water. So there's more salt in there than there is water. And so that you're going to have a lower salt concentration. So like looking down here, if we look down here and we say, okay, these dots, these are going to be the solute. So we'll pretend like that's salt. Okay. So if you look, there's a lot of salt over here. So there's not as much water over here, if that's what we're saying. There's more water over here because remember, the salt's taking up space on this side. There's more space for water to be on this side. So what's happening is water is going to go from the direction of high concentration of water to the area of low concentration of water. So what's going to happen in this example, if we have a semi-permeable membrane that doesn't allow salt to go through, it just allows water. It selects for water. It doesn't select for salt. The water is going to go to this side. And so if we look here, the same concentration is going to be on both sides. Now, there's more salt over here, But if we think about it this way, if we were to like maybe taste the water and see, it'd be just as salty on this side as this side because the ratio of salt to water is the same. So also something to remember about salt's a solute, so that's the little dots. When it's concentrated inside or out of the cell, water is going to move one way. So that's why if you eat a lot of salt, you're going to feel thirsty because it's pulling water out of your cells, and your cells need water. Okay. If we're looking at this example, we're saying kind of the same thing. This is just a little different view. So if we can tell, well, there's less, if we're talking about the ratio of sugar to water, there's more sugar over here and less water on this side than there would be on this side. So water's going to move this way to kind of dilute it and make the same ratio of sugar to water. Now, this is a case of osmosis because water's moving. If we're talking about up here, water's not moving. Other things such as glucose is moving. So diffusion and osmosis, these are both types of passive transport. These are types of passive transport. And what we mean there is we mean it uses no energy. So things are natural. If the way that I think about it is things naturally want to spread out and have enough room. So we could talk about people, we could talk about cologne, we could talk about anything like that. Things want to naturally spread out. And it just naturally happens. We don't have to put energy into it or force it to happen. It just happens. So it's kind of like going with the flow. Sometimes we say it goes down the concentration gradient, it goes with the flow. It doesn't require energy. Now, if we're talking about osmosis and we're talking about water moving one way or another way, we'll talk about the solutions. So if we're talking about cells and we're talking about the type of solution that they're in or the type of water that they may be in, we'll talk about the different type of solutions. So first thing we have, I'm going to move this down here. Actually, no, I changed my mind. I'm going to come up here. First thing we have is something called an isotonic solution. Okay, solution is isotonic. We'll leave that. So iso, if you think maybe you've watched some basketball or know basketball, sometimes they talk about running isolation plays. Or if we're talking about a football play, you might have an iso run. Well, what that means is you have the same amount of people on offense and defense. So a lot of times we talk about like iso ball. That's maybe LeBron James, and if we're talking a basketball player, maybe LeBron versus one other person. It's like 1 versus 1, the same amount on offense or defense. That's why they call it an iso play. Like with football, if we're talking an iso run, well, you have the same amount of people on each side of the line. Okay. Now, if we have the same amount of water, so let's look at this example. So we said this is a 10% salt solution, and inside the cell is a 10% salt solution. So that means the other, if we were talking about 100%, 90% of this is water, 90% of this is water. So remember, we said water goes from high to low. So there isn't a high, there isn't a low, but water is still moving because water, things just kind of move around. If a good example of this is if I were to take a jar and I were to drop food coloring in it, I would drop food coloring in it, but even though I'm not moving the water around, the food coloring would spread out. That's because the water is moving around. We don't see, it doesn't look like it to the naked eye, but it is moving around. And what's happening is eventually that food coloring is going to diffuse. It's going to spread out in that jar of water, that bottle of water. So water's going to go in and water's moving. Water's going to go in and out at the same amount. So the cell isn't going to go get bigger, isn't going to get smaller because water's going in and out at the same amount of time, or it's going in the same amount, I guess. The next thing we have is this case where we have more salt inside than outside. So if we pretended, okay, 10% salt, that means there's 90% water out here. 80% salt, that means there's, or 20% salt, that means there's 80% water. So 90% water on the outside, 80% on the inside. So it's going to go high to low. And so what happens is water is going to go inside the cell. So this is going to be a hypertonic solution. Or I'm sorry, a hypotonic. This is going to be a hypotonic solution. So hypo, Hypo, that means less or below. So hypo means less or below. So if we're talking about, you might have heard of hypothermia. What that is that's when people get really cold and their body temperature is below normal. So hypo means less. So in that case, it'd be less than the normal temperature. So if we're talking about cells, and in this case, you're going to have less solute, less salt on the outside than inside. And what that does is that causes water to go inside and gain. The way that I remember this is the cell, water's going inside the cell, and so the cell is going to get bigger. So the cell's going to grow larger, if we look here. And the way I remember it is this kind of sounds like hippo. Hippos are big. So if the cell gets big like a hippo, it is a hypotonic solution. That's the way that I remember it. On the flip side, if we have water going out of the cell, so we have 90% water inside, 20% salt, that means there's 80% water on the outside. So water is going to go on the outside. This is going to be a hypertonic solution. So what's happening here is we have more solute, more of the salt or whatever these dots are going to be on the outside than on the inside. So that's going to cause water to go out. So hyper, Hyper, that means more or above. We can kind of think of it that way. And if you think about somebody that's hyper, they're like bouncing off the walls, you know, to just have a ton of energy. Well, what we're saying is they have above normal energy. That's why they're like bouncing off the walls. Okay. With this case, water is going out of the cell, so that means it's going to shrink. And if the way that I remember this is those hyper kids, those kids that have so much energy and are bouncing off the wall, it always seems like they're like super skinny and like they burn off all their energy. And so they're small, it seems like. So with this one, the cell's going to get small like that hyper kid is. Okay. Now with plants and animal cells, if they swell up and they shrink, that can mean different things. In animal cells, that if they swell up, they can burst, and that would cause the cells to die. Plant cells, they'll swell up, but they won't. It takes a little longer for them to burst because they have something. They have those cell walls. Plus, they have these contractile vacuoles that will pump water out of the cell to prevent this. Animal cells do this too, but if the concentration is too great, they can swell up and burst. Now, if we're talking here, if the cell shrinks too much, it could die. So in plants, you know this as wilting. And animal cells, it'll shrink up and shrivel up. So this is why it's dangerous to drink seawater in this case. And if you look here, we talk about that. It's not necessarily going insane. It's actually going to cause you dehydration. So hopefully so you guys should watch this, get your notes filled out because of this. If you need to go back or something doesn't make sense, go back, watch a part of it, and we will cover active transport later.